This lecture is on RNA, information and heredity. We've discussed DNA, the structure of DNA, the function of DNA, and now we will determine the role of RNA in protein synthesis. The role of RNA. RNA is made up of, it's referred to as ribonucleic acid. And ribonucleic acid is similar to DNA, and it's made up of 3 subunits, and the subunits are a sugar, a phosphate, and a nitrogen base. So genes are made up of instructions coded into DNA. Remember we said DNA holds part of the recipe for protein, and DNA carries a code that is necessary for the production of protein. The issue is DNA cannot leave the nucleus, so it needs something to come and get the code from DNA to bring it to the ribosomes where proteins are made. So RNA is responsible for this. RNA takes these instructions from DNA and copies them to make a protein. This type of RNA that's produced from DNA is called messenger RNA because it takes the message from DNA out to the ribosomes to produce proteins. The structure of DNA includes, like I said, a sugar, a phosphate, and a base. The sugar is different from DNA because the sugar in RNA is called ribose. The phosphoric acid is the same, and then the nitrogen bases are slightly different. The nitrogen bases for RNA are adenine, cytosine, guanine, and a different base is called uracil. So uracil is replacing the base called thymine. Comparing DNA and RNA. Remember, DNA is called deoxyribose, deoxyribonucleic acid, and DNA contains the sugar deoxyribose, where RNA contains the sugar ribose. The structure of DNA is double-stranded. You remember seeing the double helix model and the ladder model that we talked about for DNA. Well, RNA is only single-stranded. It's basically 1/2 of what DNA looks like. The bases, RNA has uracil instead of thymine, so you won't see thymine in RNA at all, and you won't see uracil in DNA. Uracil replaces thymine in RNA. DNA can only stay in the nucleus, so DNA is going to need a messenger to carry the code that it produces to the ribosomes to make the protein. So RNA will be doing that. So when we produce this RNA, we call it messenger RNA. And producing this type of messenger RNA is called transcription. So RNA will take that code from DNA and move out of the nucleus. The complementary base pairs in RNA are a little bit different than DNA. RNA is adenine and uracil, and cytosine and guanine. Functions of RNA. Basically, as we talked about, RNA is going to take the code from DNA because DNA can't leave the nucleus. That's why it's called messenger RNA, because it takes the code from DNA in the nucleus to the ribosomes. RNA is responsible for making proteins using this code. This process is called transcription. So far we've talked about replication, which is making DNA. The second process we're going to talk about is called transcription, and we're basically transcribing or copying the code from DNA to RNA. That's why it's called transcription, to make messenger RNA. Making the protein is called protein synthesis, or another word for protein synthesis is called translation. So we have replication, is copying DNA, transcription is creating messenger RNA, and protein synthesis, which is translation, that's creating a protein. There are 3 types of RNA. We have messenger RNA, ribosomal RNA, and transfer RNA. Messenger RNA carries information from DNA to the ribosomes to make protein. Ribosomal RNA makes up the ribosomes, and transfer RNA carries amino acids to the ribosomes to make the protein, because remember, the building blocks of protein are amino acids. Okay, producing RNA, again, RNA synthesis is called transcription, and this is going to happen in the nucleus because to make the RNA, we need to use the code from DNA, and DNA cannot leave the nucleus. So RNA synthesis or transcription takes place in the nucleus. The DNA unzips with the help of an enzyme called RNA polymerase. RNA pairs up with the complementary bases in DNA. So the base adenine in DNA would pair up with uracil to make RNA. And cytosine would pair up with guanine just like in DNA. DNA is used as a pattern or a template to make RNA. So for example, if the left half of the DNA looked like this, CGATTC, what would the right half, or what would the RNA look like that's made from this DNA? C would pair with G, G with C, A usually pairs with T when we're making DNA, but we're making RNA, so A will pair with U, then T is A, and C is G. So hundreds and thousands of copies of RNA can be made from just one single gene or strand of DNA. So we have what are called promoters on our regions of DNA, and they have, on our regions of RNA, I'm sorry, they have specific sequences on DNA that RNA will copy. So this RNA polymerase, where to start and stop making RNA, there are certain parts of the RNA that will say start or stop. So in order to determine where on the DNA or the RNA we would like to create RNA using transcription, we have a start codon. So there's a certain sequence of bases on RNA that will tell us this is where we want to start making the protein. So RNA molecules sometimes require bits and pieces to be cut out of them before we actually pick up those amino acids to make the protein. Those portions that we cut out and discard are called introns. 13.2 talks about ribosomes and protein synthesis. So the first step is to use the code from DNA that is transcribed to messenger RNA. So the process of transcription, now we have our messenger RNA, which is carrying that code from DNA and has to carry it out to the cytoplasm during the process of making proteins. DNA hold part of the recipe for making the protein. So protein is made up of a specific sequence of amino acids. So on our, you can see at the bottom of this slide, there is a strand of RNA, and we break that strand of RNA up into 3-letter words, which we call codons. And these codons are 3 bases that are going to specifically code for a single amino acid to be added to this polypeptide chain. So the polypeptide chain is actually the long strand of amino acids that make up a protein, because proteins are referred to as polypeptide chains as well. So we take our long strand of RNA that was made from our DNA and we break it into groups of 3 bases, which are called codons, and those codons will code for one single amino acid, and then we connect those amino acids. So how do we tell which amino acids are picked up from those codons? We're going to use what's called a codon chart. So you can see on this slide we have a codon chart. You're going to see in the middle we have a G, a U, an A, and a C. And then the first circle, outside that middle circle, you see all those bases listed. And then there's another darker yellow circle on the outside, and then you see more of the 4 bases listed. And then the very outer ring, you see the amino acids that are listed. So let's go over how to read and use this codon chart. This codon chart is a messenger RNA codon chart, and we have to use our codons from messenger RNA to determine which amino acids are picked up. So there are 4 different bases in RNA. We have A, G, C, and U. We're going to use the center letters, which you see are the G, U, A, and C. Then we're going to go to the middle ones, which is a little bit darker yellow. Then we go to the outer ones, which are the darkest yellow. And then that will tell us which amino acid is actually picked up. So if I wanted you to code, let's say, CAC. So I'm going to start in the middle with my 4 bases, and I go to C, C is in red. So that's my first one. Then my second nitrogen base is A. So I'm going to go to my second ring, and you see A that's posted there in red. So far I have C, A, and then I'm going to go to my third base, which is another C. So you see it in red there, CAC. And the amino acid that CAC codes for is called histidine. So you always start in the middle with the light yellow. Then that determines which piece of the pie, so to speak, that you will continue decoding for which amino acid gets picked up. So here we see, you see how it's separated into 4 boxes. We're going to go to that bottom lower right-hand side box because that's where C is. Then I'm going to go to my second ring, which is darker yellow, and I have to find the next base, which was A for this particular decoding sequence. So I had C, and then I go to A, so I find A, and that shows me the next piece of the pie that I'll continue with. And then my last nitrogen base is C, so then I find C in that piece, and I end up with histidine. So histidine would be the amino acid that is picked up for the codon CAC. Okay, translation. The order of bases in messenger RNA, those order of bases that we got from DNA, coded from DNA, gives the order in which amino acids should be joined to produce a protein. Ribosomes use a sequence of codons in messenger RNA to pick up specific amino acids, which we just did using our messenger RNA codon chart. And then the decoding of messenger RNA message into a protein, that whole process is known as translation. So we started with DNA that had a code. RNA went and paired up with that DNA according to whatever that code was. Then RNA, we used the codes from RNA in that codon chart to determine which amino acids get picked up. We put those amino acids in order, and then that long chain of amino acids gives us a protein. That whole process is called translation. So translation is going to occur in the nucleus, and it goes through that whole process of DNA to messenger RNA to that codon chart to picking up an amino acid. And one at a time, the ribosomes are then going to attach each amino acid and make this growing chain, which will end up to be a protein. Okay, the protein continues until the ribosomes reach what's called a stop codon on the messenger RNA molecule. So if you go back to this slide and you look at the very outer ring where the amino acids are, you're going to see the word stop in the upper right-hand quadrant. It says tyrosine stop, cytosine stop. Okay, so that is a codon that's specifically coded for the word stop. That tells us where the protein is going to stop. So when you get to that stop molecule, it's going to release both the newly formed polypeptide and the messenger RNA molecule, completing the process of translation. So the codons of messenger RNA specify the sequence of amino acids in a protein. Proteins, in turn, play a key role in producing an organism's traits. So there is a link to a video at the bottom in the box. And what I want you to do is copy and paste that video. It's a 2.5-minute video. Copy and paste it into your browser and watch the video. And it will pretty much summarize through a video everything that I just talked about. And that is it as far as we're going to go for 13.1, which is